Chapter 10, Section 3 talks about DNA replication. And when DNA is replicated, here's our strand of DNA. And the helicase molecule, which is an enzyme, again that ACE ending generally signifies that it will be an enzyme, is going to run along the strand of DNA and break the hydrogen bonds that are holding together the bases. That will allow the chains to separate, and now we'll have two separate chains to be able to use as a template for building a new strand with that old strand. So at the end, we'll have two new pieces of DNA, each made up of part of an old strand and part of a new strand. The part where they are split, this section right here is called the replication fork. So just as if you were to get to a fork in the road, you have to go one way or another. The same thing is happening here. You're going to one strand or the other side. DNA polymerase is another important enzyme, and the helicase will split the DNA double helix, and DNA polymerase is going to be the molecule that will attach to each of those separated strands of DNA and assemble the complement to it. So like in this diagram here where we're separated out, this polymerase molecule will travel along. If there's a G here, that's going to pair up with a C. If there's a C, that's going to pair up with a G. And it's going to continue making that new strand as a complement to the old strand. The same thing will be going on the other side. And there are actually thousands of these polymerase molecules that are going to start at various parts along the strand of DNA so that they each can do a segment, and you don't have to wait for one molecule to go all the way down the entire strand of DNA. So I would expect you to know the base pairing rules, and if I give you an old strand, you should be able to tell me what its complementary strand would be. And finally, this process needs to be very accurate. If it's not accurate, then that will cause a mutation, which is just a change in the nucleotide sequence. So that would be like a natural mutation if it happens through this replication process. But these mutations can also be caused by outside factors like chemicals, radiation, UV light. All of those things can also contribute to mutations, but we try to limit the natural ones, such as through this replication process. So there's actually a really high rate of accuracy, and there's usually about 1 in every 10,000 bases that there is an error. So to combat that, there are also enzymes that will proofread these strands of DNA, and if they find a mistake, they can call in a repair enzyme that will come and fix it. So once the proofreading and the repair part is done, then that limits the error rate to about 1 in a billion. And so there's really a high rate of accuracy with this replication process.